Today we will discuss about what are the factors that affect the extracellular matrix stiffness. The factors here we will discuss with related to the tumor cells. So how these tumor cells can affect the stiffness of the extracellular matrix. So this tumor cells uh, has some reprogramming that has occurred within the tumor cells, and that will lead to the secretion of the cytokines, and that helps in the remodeling of the cellular matrix. For example, this this TGF beta receptors. TGF beta is usually cause the activation of this cancer associated fibroblast, and at the same time what it will does is tumor the cancer cells also promotes the collagen cross linking. So collagen cross linking, and at the same time this activation of cancer associated fibroblast will ultimately leads to the tumor stiffening, and at the same time excessive deposition of this ECM component around the cancer cells, and this will act as a physical barrier that will prevent the tumor cells from the immune cells to come and detect this tumor cells, or from the immune response. To prevent the tumor cells from the immune response as act as a physical barrier, apart from the other factors such as glycocalyx that is also been dysregulated in the case of tumor cells. So this is just a brief overview. Now we will understand in slightly in detail how this increased stiffness will further uh, promote the uh, cancers cancer cell phenotypes. So one thing that will happen within the cells is this change in the reprogramming is that this aerobic aerobic glycosylation occurs, not glycosylation, aerobic glycolysis occurs, and that will lead to the formation of this glucose to pyruvate without the need of the oxygen, and that will cause the more lactate formation. At the same time, this nucleotide metabolism will also been produced during this production of glucose to pyruvate. So these things will get upregulated in the tumor cells that has been mostly observed, and this thing will help the cell, since now the cells is consuming more glucose. Instead of going to the mitochondria, it will directly convert glucose into the pyruvate. So this will uh, accelerate the rate of growth, or it will lead to this proliferation of the cells. And the other things, these tumors, so this reprogramming is occurs because of the ECM stiffness, and both of them are interrelated. That is, first this tumor cells will cause the change in the composition of the ECM, and the change in the composition of ECM will further aggravated the reprogramming within the tumor cells, and that will cause further increase in the stiffness of the ECM. So this is the you can say that work in a feedback loop or in a cascade manner. So the and the other changes that occurs in the cancer cells are in the formation of the shape, because and these things is occurs because of the force exerted by the extracellular matrix. That is, they will form these pseudopodia formations, or spreading area, or they can attach to the other cell surface, or these cell adhesions will be changed. And this proliferation, the excessive proliferation will lead to the exosome secretion, and also this stromal cell autophagy. So this will cause these two conditions, and that is also works in the favor of cancer cells progression. And because of the stiffer ECM, what it will cause is it will reduce the possibility. It will reduce this drug delivery possibility directly to the tumor cells. Also, this drug efflux, that is to uh, prevent the to export the drugs without to export the deflect. You can say that deflect the drugs, and prevent it from being prevent it from being detect prevent it from we can say it in a different term, such as drug effects is work in a way that acellular matrix will prevent the drug to come and interact with this immune cells, or not in come and interact with these cancer cells. Okay. And the next part is that this immune cell infiltration, because of the physical barriers of the stiff ECM, the immune cells also will not be able to infiltrate the ECM and act upon these cancer cells. And since this immune cells will has many checkpoint inhibitors, cytokines, and that will inhibit the immune cells to come and act on the tumor cells. And this we have covered many times in our previous videos. You can refer those previous videos. So this is just a detailed overview of this immunotherapy that comes in this two part. First one is this immune checkpoint blockade. So one thing that can be done is we can block the checkpoint on the immune cells, so that this immune checkpoint therapy will not be the immune cells will not be deactivated by the cancer cells. Other things that can be done is this CAR T cell therapy. That is, CAR T cells has specific antigen information about the cancer cells. Next one is this oncolytic virus therapy. So this will act upon those tumor cells where there is a reprogramming has been done. So it will not act on the healthy cells, but only on the tumor cells. And the next one is this the therapeutic cancer vaccines. In this case, the antigens, inactivated antigens, is been introduced into the cells that can help the immune cells to develop antibodies against the antibodies that is specific to the tumor cells. However, this efficiency will also be reduced because of the stiffer ECM, as the cells will not be able to come and infiltrate the tumor cells. And in case of this drug delivery reduction and drug efflux, therapy that is commonly been affected because of these factors are this chemotherapy resistance. So in this case, what will happen is that the similar case, because of the physical barrier, the chemotherapeutic agent will not be able to act upon this tumor cells. And this stiffer ECM will promote the reprogramming in the cells in a way that it will help in this ECM. It will help in this ETM for ETM transitions, that is epithelial to mesenchymal transition, that has been shown here. And this epithelial to mesenchymal transition will help the cells to spread from origin of the cancer cells to this different organs. And before, although we have covered many times in our previous videos, we just you can see the main point in this slide. That is, first, the invasion of the cells occurs because of the ECM. It will promote the more EMT characteristics, and thereafter, once it is in the blood vessels, it will when it reaches to the site of the origin. This is right now it is not the blood vessels. This is the blood vessels. So first the invasion occurs. It will lead to the epithelial cells, primary tumor site, and then it will go to the intravasculations. Now it is in circulations with blood, and when it reaches the secondary organ sites where it can infect or spread these tumor cells because of the epithelials, lectins formations, and these cancer cells also has a lectins that can interact with this epithelial cells on the metastasis site. There it will help in the extravasations, and then the colonization occurs, and transition from EMT to MET will finally leads to this colonization of the tumor cells at the metastasis site. So this is one example that can be done from the metastasis site, that is from this part to this part, or from liver, or from this pancreas to this liver. So this is how the ECM stiffness will promote the cancer cell phenotype. And we have also discussed in brief what are the factors that affect from the pancreatic cancers. What are the factors from the pancreatic cancers that will lead to the change in the ECM composition or increase in the ECM stiffness. So I hope you got a basic idea about this relationship between ECM stiffness and cancer cell phenotype. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.